Good morning, all of you, and welcome to this class. Today, I will talk about development of male gametophyte and female gametophyte. First of all, see the structure of pollen grain or microspore. In previous lecture, we already discussed about the process of microsporogenesis. In brief, say here the pollen mother cell undergoes meiotic division and they form pollen tetrad. The pollen tetrad means all the four pollen grains join together and form a tetrad. Usually, the mature pollens are not attached in tetrahedral. They get separated from one another. See here, all the pollen grains are separated from one another. Then say, look here, the single pollen grain. The morphology of pollen grain is the pollen grain is the first cell of male gametophyte which contain only one haploid nucleus. The wall of pollen grain is made up of two layers, the outer exine and inner intine. The exine is tough, warty and spiny. It is highly resistant layer. It can protect the pollen grain from external injury. The exine is made up from sporopollenin substance. The exine is in some places is thin and that area of the portion it is called the germ pores. Below the exine, single layered very thin intine is present. It is elastic in nature and delicate. It is made up of cellulose substance. Look the character. Microspore, that is pollen grain, is the first cell of male gametophyte which contain only one haploid nucleus. The exine is cutinized and tough with different ornamentation. It may be warty, spiny. It can protect the pollen from external injury. The exine is made up of fatty substance called sporopollenin. The intine is very thin, elastic and delicate and made up of cellulose substance. So this is a simple morphological character of pollen grain. Let us talk about the development of male gametophyte. The development of male gametophyte process occur in two places. That means the before pollination means inside the anther, and second after pollination, that means on the stigma. As I said earlier, the pollen grain is the first cell of male gametophyte which contain only one haploid nucleus. The haploid cell undergoes cell division and they form two unequal cells, smaller generative cell and larger vegetative cell. The generative cell initially attached with the intine and the larger vegetative cell is in the cytoplasm. Later on or very soon, the generative cell is separated from intine and freely float in the cytoplasm. In this stage, the pollen grain is two-celled. This two-celled pollen grain is released from the anther after dehiscence of the anther wall, and these two celled pollen grain is landing on the stigma. Then the pollen grain is absorbed nourishment or watery sugar from the stigma and pollen tube. Ultimately, the pressure will exert it or increase on the anther wall. And due to the increase the pressure on anther wall, the germ pore is ruptured or break, and the inner layer in time is started elongation or extend, and they form the pollen tube. Later on, this generative cell and vegetative cell both are migrate along with the cytoplasm in the pollen tube, and at the base of pollen tube form the callus plug. What is the role of callus plug? The callus plug is prevent the backflow of cytoplasm, and later on, then the small generative cell are divided and to form two male gamete. In this way, the male gamete is developed. This whole structure, it is called the male gametophyte. See the character of developed male gametophyte. Usually, the mature pollen grains are not attached in tetrad, and they get separated from one another. Microspore, that is pollen grain, is the first cell of the male gametophyte, which contain only one haploid nucleus. During early stage of development, it remains within the microsporangium. The cell undergoes unequal division and form a small generative cell and large vegetative cell or tube cell. Initially, the generative cell remains lying at one corner of the spore wall. At this stage, the pollen grain is two-celled. A large vegetative cell and a small generative cell. The pollen grain may be discharged from the anther at this stage, that is the two-celled stage. The liberated pollen grain germinate on the stigma and produce pollen tube. The pollen tube absorbs nourishment and sugary fluid from the cell of the stigma and pollen style for its growth. The tube nucleus migrates to the tip of pollen tube. The generative cell also passes into it and divides inside the pollen tube to form two male gametes. Let us talk about the structure of pistil. Sometimes we call gynoecium. This is the female part. It is the central or topmost wall of the flower. A pistil consists of three parts: stigma, style, and ovary. Stigma is rounded head, is a specialized part of the pistil on which the pollen grains are landing. Style is the tubular structure that connect the stigma with ovary. And third one, the lowermost solan part is the ovary containing one or more ovules. It may be monocarpellary or polycarpellary. So the character of gynoecium. The gynoecium or pistil is the central are topmost whorl of the flower. A typical carpel has three parts: ovary, style, and stigma. The stigma is the specialized part of the pistil on which the pollen grains are trapped during pollination. The style is a tubular structure that connects the stigma with ovary. The lowermost solan part is the ovary containing one or more solan bodies called ovules. So these are the character of gynoecium. Then we talk about the megasporogenesis. So what is megasporogenesis? The formation of haploid megaspore from diploid megaspore mother cell, known as megasporogenesis. See in detail, the archisporium cell. Archisporium cell is the hypodermal in origin. What is the hypodermal in origin? The one cell of ovary wall with dense cytoplasm and prominent nucleus, that is the archisporium cell. That archisporium cell active and divide periclinally, and they form outer parietal primary parietal cell and inner primary sporogenous cell. Outer primary parietal cell may or may not be divided further. Inner primary sporogenous cell does not divide it further, but directly working as a diploid megaspore mother cell. See here in diagram, this is a diploid megaspore mother cell. This megaspore mother cell undergoes meiotic division and they form haploid 4 megaspore. They are arranged in a linear tetrad. Out of 4 megaspore, 3 megaspore is degenerated and only 1 megaspore is functional. Look here, this is the functional megaspore and 3 megaspore are degenerated. In theoretical portion, the archisporium is a hypodermal in origin. The archisporium divides periclinally into outer primary parietal cell and inner primary sporogenous cell. The primary parietal cell divides further, or sometime it remains undivided. The primary sporogenous cell usually does not divide it further and functions directly as megaspore mother cell. The megaspore mother cell now undergoes meiosis or reduction division, and usually a linear row of 4 haploid megaspore cells is formed. Out of 4 megaspore, only 1 megaspore is functional, while other 3 megaspore degenerated. See in the diagram, this is the single megaspore mother cell which is diploid in nature. They form a 4 haploid megaspore arranged in linear tetrad in a row. Out of 4 megaspore, 3 are degenerated and only 1 is functional. Look here, all 3 megaspore is degenerated and only 1 megaspore is functional. This is cellular diagram. The primary archisporial cell divides periclinally and form primary parietal cell and inner primary sporogenous cell. The outer primary parietal cell may be divide or may not be divide further. The primary inner sporogenous cell directly working as megaspore mother cell, and and it undergoes meiotic division and they form four megaspore, haploid megaspore. Out of four megaspore, three megaspores are degenerated and only one megaspore is functional. See here, this is the functional megaspore and three megaspores are degenerated. Later on, this haploid megaspores undergoes first meiotic division and they form two-celled stage. These two-celled stages again undergoes second meiotic division and they form four-celled stage. Again this four-cell undergoes meiotic division, means it is undergoes in third meiotic division and form 8-cell stage. Eight-cell stage. So the four nucleus out of eight nucleus, the four cells are arranged on one pole and four nucleus are arranged on the different pole. Means the four cell arranged in micropylar end and four cell or four nucleus are arranged on the chalazal end. Later on, the one nucleus from each pole is migrate towards the center and they form diploid secondary polar nuclei. Look here, this is the secondary polar nuclei and the remaining three are arranged on the different poles. Means the three are arranged on the micropylar end and three are arranged on the chalazal end. The chalazal end three nucleus they form antipodal cell and the three nucleus of micropylar end they form single egg apparatus present in the center and surrounded form the synergids. So in the micropylar end the haploid cells are formed, which is single egg apparatus present in the center surrounded by two synergids. Inside the synergids, finger-like projection is present, that is called filiform apparatus. And what is the role of filiform apparatus? To guide the pollen tube during fertilization or during the entry of pollen tube in the embryo sac. And centrally present the secondary polar nuclei, which is diploid in nature. What is the role of secondary polar nuclei? It take part in the fertilization, that is the double fertilization, and this is the important characteristic or unique characteristics of angiosperm. And three antipodal cell, haploid antipodal cell arranged on the chalazal end. In this way, 7-celled and 8 nucleated embryo sac is formed. See in theoretical portion, in angiosperm, the development of female gametophyte is completely endosporous, that is within the megaspore. The functional megaspore divides into 2, then 4, and finally 8 daughter nuclei, 4 of which are located at each pole. Then one nucleus from each pole moves to the center of the embryo sac and fuse, they are forming the secondary nucleus. Finally, the embryo sac or the female gametophyte become organized. In typical embryo sac, it is 7-celled and 8 nucleated structure. At the micropylar end, a group of 3 cells are present called egg apparatus. It is composed of one egg cell and 2 synergids. At the chalazal end, another group of 3 cells are present called antipodal cell. One nucleus from each pole moves to the center of the embryo sac and fuse, there forming the secondary nucleus. This is the only diploid cell in the embryo sac, while other cells are haploid. That is, the centrally present secondary polar nucleus is diploid, and the remaining micropylar end egg apparatus is haploid, and chalazal end antipodal cell is also haploid. Now see, the one megaspore is functional and 3 are degenerated, means the only one functional megaspore participate in the development of embryo sac, that is called the monosporic ovule. In this way, if the 2 megaspore functional and 2 are degenerated, means the 2 megaspores are involved or participate in the development of embryo sac, that is called as the biosporic ovule. 3 megaspore functional and 1 are degenerated, that is called trisporic ovule. And if all the 4 megaspore functional and none of these degenerated, that is called the tetrasporic ovule. So this is the whole process of female gametophyte, development of female gametophyte. Thank you. If you have any question, then you can ask.